All right. Hello. Welcome back to my channel. Today we've got a video on an interesting use for AI that I found today. So this morning I saw a really interesting video, something I wanted to learn more about. It's called DNA and RNA vaccines. So RNA vaccines are a recent breakthrough, but like with any other technology, there are risks to be aware of, and there's also benefits. And so we want to know about that. But I thought this was really interesting, and the person being interviewed is Dr. David J. Spiker. Super intelligent guy, extremely bright, and his resume is very impressive. And I don't have a medical background myself, other than a few medical mission trips and a pet interest in health. And so I like to keep up with these things. But one of the issues I ran into is that unfortunately, Dr. Spiker here has some sort of speech impediment. And so, and I actually have this on 2x speed, and I found that made it a little bit easier to follow the conversation, but it was still something I felt like was taking away from the overall delivery of his message, unfortunately. And so I tried to think of a way I could use AI to solve this. And so if I were the owner of this original MP4 file, I had the video and the audio, I could upload this to an AI software and I could have it cut out fillers, and I might even be able to keep the video, and it would cut out, maybe the gaps where there's stuttering occurring or pauses. And, if a human were doing that, that would take an enormous amount of time to do that type of editing. But I don't have that, so I can't really try that approach. And downloading this is not very practical right now. But this, to me, was a great opportunity to explore the uses of Notebook LM. Now, this is a really great um, tool here where you can upload certain sources, and then what the AI is doing is a sort of retrieval augmented generation function, which we will refer to henceforth as RAG. And essentially, what it's doing is AI has all of its knowledge, but it's so much knowledge that it's hard for it to be precise. And so, when you give it certain sources or materials, you give it a sort of lens or a filter to pull information through to know what it is more specifically that you're interested in or what you are looking for. And so what I did here is I created a new notebook. The first thing it'll ask is like upload sources. So I'll go here and I will click on this, copy the link, bring it to Notebook LM. This is a YouTube video, and we're going to run into a problem. Spoiler alert. So there's the copied link, and there it is. So this video cannot be imported. Transcript is not available. Now, that's actually not true. The transcript is available. We have it right here, so I don't know why YouTube is doing that. But what we can do then is, this is a different one. Yeah. So I can copy this, scroll down, and I'm getting these numbers in, but AI is so smart, it's going to know these numbers don't really have any purpose other than the order of the conversation, and it's not going to confuse it. Okay, so don't worry about that. All right, so copy this, go back to our notebook. Oh gosh, am I fumbling? There we go. All right, so source, and we're going to paste a copied text. This is going to be our transcript, and voila. So now from here, what's really amazing is this is really crazy to me. Podcasts can be a great way to get information, especially when the guest is very knowledgeable, like Dr. David Spiker is. But what this offers you that normal podcasts don't is you can actually engage with the material. So one of the questions, and I should probably talk about his study. They looked at in Canada, they looked at 30 different vials of RNA vaccine, something like 30, I don't know, 27 to 38, somewhere between there. And they found DNA contamination. And now DNA is not supposed to be in there. And when it gets into the cells, the RNA is supposed to serve as a blueprint, give instructions. It's more temporary. But the DNA being in there can cause more long-term problems. And especially with the use of the lipid nanoparticles, that allows for this problem to become more exacerbated because now the DNA contamination is much more far reaching throughout the body. And so it can cause problems in many different areas. otherwise not would have happened. So anyway, this is a very interesting problem to be exploring. But now that I've uploaded the transcript, I can actually kind of engage with the material. I can ask the questions as though I'm asking Dr. David Spiker himself. And I could even ask it to generate multiple choice questions. We might play around with that. Let's go ahead and say, create three multiple choice questions based on the material. But yeah, and I could also just ask questions like the ones it was prompting, because like what are the risks? What are the harms? Yeah, so this is really cool already. Really cool. Let's ask it, how does the SV40 enhancer potentially affect the integration? Um, Now, the SV40, they talk about it in the podcast, is not actually something that the manufacturers think are a concern, but it's the position of Dr. David Spiker and others that it's premature to make that call, um, because SV40 has been known to cause certain problems in other mammals. All right. So great. You're already kind of like, this would be great for pre-learning before you get into the information. But let me get back to the main thing. So there's also a feature here where you can ask it to generate a podcast. Notebook guide. There it is. So notebook guide is going to pull up this HUD. And here I get a summary of the interview, but also I could ask it to create a podcast. Now, this is really great for this situation where Dr. David Spiker's speech impediment makes it somewhat difficult to follow along, unfortunately, while I may be multitasking or something like that. And so I can click generate, and this is going to take a while. It takes probably 5 to 10 minutes, but I've already done the work in advance of this video. And so here's what I got. All right, so today we're doing a deep dive. And let me crank the speed down because not everybody's crazy. All right.
Into some findings about RNA vaccines. And with us to help us unpack this is virologist Dr. David Spiker.
Thanks for having me.
So RNA vaccines, right? Something we've obviously heard a lot about, but specifically we're diving into some discoveries around DNA contamination.
So there you have it. I listened to this full thing already. It's really great. The last minute and a half is dedicated to a sort of recap. And some of the things they're talking about, your first thought or concern with AI is this thing hallucinating? Is it making things up? And so as you are listening to this 10-minute version as opposed to the 1 hour version, you can sort of hear certain topics get brought up. And one of them was, they talked about ribosomal shifting. And so I have the transcript pulled up and I can kind of control F and see ribosome. So here we have one mention of ribosomes, and now here's the mention of ribosomal shifting. So if I wanted to learn more about this, I hear it mentioned in the podcast, is that a hallucination? Is that legit? I can actually go further into that. I can go to 4416. I know not only do they actually talk about it, but what are they saying? And does it line up with what the AI is saying? And so far, based on what I have listened to of this and what I have and the entire podcast that Notebook generated, it's pretty spot on. I wonder what's lost in the fact that it's, only 10 minutes as opposed to 45. And I think a lot of what's lost is it talks about his specific research in the 30 something vials that they studied and analyzed and saw that DNA contamination in 100% of them. It didn't talk about it at all in the Notebook podcast. But so yeah, that's unfortunate, but still that's something you can see pretty early on if you're just kind of scrolling through. Where is it? Yeah, this is his article or publication if you're interested. And yeah, 38 vials. Okay, so I was actually wrong. I said 20 something to 37, 38 vials. So anyway, this was not mentioned in the Notebook podcast, unfortunately, but I think it's kind of looking at everything as a more broad view, and it's talking about the findings, and it didn't really go into the methods. So maybe it'll get better in the future. What else? I'm trying to think. Oh yeah, so it talked about ribosomal shifting, and I thought that was great. Another thing they mentioned in the podcast, I'm wondering, are they just making this up? They talk about there being a lack of government funding for independent research into the efficacy or safety of these things. And so I can again go to the transcript, talk about, let's find one of these keywords, and AI will use a lot of synonyms. It's almost like a high schooler trying to avoid plagiarism. It'll just like switch out words, haphazardly or synonyms to get away from saying, to get away from quoting. But anyway, funding is a key word that it couldn't get away from. And so, yeah, governments aren't funding this sort of research. I've been out of a full-time job. Um, Tournaments aren't funding. So, yeah, there you have it. And so I would recommend when you're using the Notebook podcast to sort of keep the transcript up with it and go from there. And then if you want to ask more questions, you can do that. And I think that's really awesome. So, what are the implications of DNA contamination? And we'll get a great answer on that, I'm sure. Maybe won't. Okay, so you got to be patient. All right, there it is. Potential health implications. The SV40 enhancer, contamination, source of DNA, increases nuclear integration, um, lipid nanoparticles enhance DNA uptake, integration to human DNA, traumatic repeat, pre-cancer gene. I think this is people's biggest concern right now. We are seeing an uptick in cancer diagnosis for young people. But that's been going on for a while. So anyway, but another cool thing is these citations it has. So this comes from 330. So like this is awesome. If I didn't want to listen to the entire podcast and I just want to know, where is the relevant information? Boom, here's like an interesting claim right here. Like I want to listen to them talk about this. So I'll click on 5. Boom, takes me right to it. And this is kind of a newer feature for AI to be able to tell you where in the RAG it comes from, where in the chunks, because that's how AI does this. It chunks it all up. But anyway, won't bore you with that. So yeah, this is a great use of AI. I'm really glad I followed this idea down its rabbit hole into its conclusion. And I hope you found it interesting as well. I can imagine a lot of uses for this. I'm probably going to make a second video where I look at somebody like a different doctor who talks about really complicated things. But the beauty of the Notebook LM, and I haven't mentioned it yet, is that the AI generated voices are not only are they really easy to listen to, but let me go back to the Notebook guide. They have a really good ability to create analogies that it's almost like it is prepared by a teacher or somebody who's really good at speaking to in layman terms. And so while this conversation is originally had between Dr. John Campbell and another PhD guy, it can be hard to follow as just a law student. But with these two that the AI creates to do the conversation, it is much more approachable and they will always add analogies, or even one of the guests plays typically the person who's uninformed, right? And so they go, whoa, that sounds important. What is that? Or SV40 enhancer, that sounds complicated. What is that? And really helpful for me. And then they use very good analogies to help you kind of reach the same understanding that they have. And so, yeah, I think AI is extremely impressive right now, and I can't wait to see what the future holds for it and how much better it gets. So I hope you found this video helpful. And that being said, I will see you in the next one. Have a good rest of your day.